everybody, I'm going to go over the wrap up for this week. I try to do that every week, or at least get all of the topics for you prior to your exam, so you'll be able to at least try to figure out what to focus on, because I know there's a lot of material. So I will also be posting some adjunct videos for you that will also help. You may use them, you may not use them, but as far as these wrap up videos, I feel this is very important portion of studying for your test. So just to go back to basics here, we are all made of cells. We start with one cell and we multiply into billions of cells. So, and there's many things, many intricate things that go on in the cells. And when we think of a cell and what goes on in the cell, it's energy. We are all made of energy in different ways. And even when you think about our heart pumping, that's also energy. So just to go over some key concepts here, active transport, osmosis, diffusion, endo, and exocytosis, just some key definitions here. So when we look at this picture here, we have to look at how things are moving across the membrane. And I'll make this a little larger here. As you can see, this is positive and negative charge. So we have more potassium in the intracellular fluid and more sodium in the extracellular fluid. And same thing with sodium and chloride attract each other, so you'll usually always see those together. But as you can see, there's still some potassium over here, just not as much. You'll see some sodium over here, but just not as much. So this is a normal looking cell, or what's going on inside the cell. When things start going haywire is when you have issues such as illness or kidney failure, as well as multiple other things, like your case study with the heart issues. Things are not balanced out, and the goal is to have things balanced out. So how do we get cells across? Well, one thing is we have to move things from a higher concentrate to a lower concentrate. So when we're moving from high to low, that is called diffusion. So you're moving from high to low. So meaning you're moving electrolytes across. Oops, high to low. And how is that done? That's done by active transport. So active transport and diffusion go together because you need energy to move from high to low. It needs to push its way. So that is ATP. And I just think of that as energy in the cell. And that helps with metabolism, all sorts of things. When we're thinking about how and what's easy, then I think osmosis. That is using more of you're going from a low concentrate to a high concentrate, and you're doing that by passive. So when I think of passive, I think of passive, it's pretty easy, you just move it on through. No need to have any extra required energy to get any of these solutes through. So that's a quick wrap up of that. So high to low is diffusion, using active transport, okay? But just remember diffusion, and osmosis is low to high, and that's using passive transport, right? Transport here. All right, so now let's go to endocytosis and exocytosis. There's a little picture here. Exocytosis is basically you have a cell, let's just say there's proteins in here, and it's moving across this semi-permeable membrane and then going out of the cell. And so it's no different with endo, it's just the opposite. So when you think of this, it's a vesicle, and it fuses with the cell membrane here. So it's moving up, it fuses with it, and then it releases all the contents outside of the cell. That's exocytosis. Endocytosis is going into the cell. The next thing I'm going to talk about is, now that we've learned about cells, and I know you had your readings and other things, as well as next week we will go more into balancing of the electrolytes within the cells. But let's talk about cells in general. Now we know we are all made of cells, and they're very finely put together, as you learned about what happens with the energy in the cells and ions going in and out of the cells, electrolytes, etc. But sometimes that can go haywire because of environmental factors, genetic factors, etc. So there's just some 4 key concepts that I want you to know about because it's very important. So I'll go to just use some examples. So hyperplasia. So we have a cell, and it doesn't necessarily mean the cells in hyperplasia change in how they are operating. But what happens is they multiply, meaning they just increase in number. And so a good example of that would be endometriosis. You have, let's just say, the uterus, and the lining becomes thicker. Well, how is that becoming thicker? Oh, this is the cervix, and here's the uterus. Not an artist. it becomes thicker. And what is that? It's hyperplasia, and all those cells are replicating. All right, so they're the same size. Now, can this turn into dysplasia here? Yes, it certainly can. They can start differentiating in size, which we'll talk about, but that's hyperplasia right here. And that's an example. It's just increasing in number. So then when we're thinking of metaplasia, that's replacing the type of cell into another type of cell. So let's say, for example, somebody with heartburn. All right, again, I'm not a good artist, but here is an esophagus to the stomach here. Okay, so let's just say, and there's a sphincter down here that lets food in, and it's supposed to keep the food in here. But sometimes this pyloric sphincter doesn't work as well, or someone's eating too much. This thing can become a little floppy, and you have food start to come up here. Your stomach is acidic. Otherwise, it wouldn't be able to digest the food that well. Obviously, there's other things that go with it, such as bile and multiple other enzymes, but this environment is acidic. This environment in your esophagus, as the food's going down, is alkaline. And there's different types of cells in the stomach versus this, the esophagus. So what can happen is you can develop metaplasia in the esophagus because if you have food, or if there's acid that's going up in here, the acid can actually change the cells in the esophagus, and that's metaplasia. Again, this can turn into dysplasia. There's precancerous cells, but that's metaplasia. It's replacing the type of cell into a different type of cell. It's not necessarily a dysplasia or a malignant cell at that point. All right, so hypertrophy is where it's larger cells. So one example for that is the heart. I'm not good at drawing a heart, but okay, that's not a heart. But anyways, let's just draw a heart like that. All right, so you have your heart, and as in the case study or the discussion, this person has hypertension over a long period of time. And when I think of heart muscle, it's a muscle. And over time, if that's being overworked with high blood pressure for a long period of time, it can become thickened, and that's hypertrophy. So when you hear left ventricular hypertrophy, the left ventricle becomes thickened, and so you can't fill the left ventricle up with enough blood because the wall is thickened here. Again, I'm not drawing this very well, but that's exactly what's happening as the cells have hypertrophy, the muscle wall has thickened, therefore you have cardiomyopathy, which is enlargement of the heart, and you can have some congestive heart failure, because if you can only fill up the heart this much here instead of all the way down here like it was before, blood's going to end up backing up. So if that makes sense. All right, now dysplasia is an abnormal cell, and it's hard to differentiate these cells. So the process, this dysplasia, you can have different degrees of it. So for example, cervical cancer, you can have a precancerous lesions, or you can have, say, a type 1, and then it can go on from there. So precancerous, you have some dysplasia. Those cells are changing, but it's not coming out as an actual cancer, but that can change to a malignant type of cell. It's not a true adaptive change, so meaning that it's not changing as it's supposed to. So a cell is supposed to change into another cell, and another cell, and another cell, and they're supposed to have normal processes. An abnormal cell dysplasia does not. You can have this, you can have this, it can look like this, which ends up, or can end up turning into malignancy if it's not treated. There's a change in cell size and cell shape, and they can be immature or they can be mature. So there's really no rhyme or reason to this, so it's not that mature cells are replaced by another mature cell. It can be immature, it can be mature, and they're just not functioning as well as they should. They're not going to work, so this can turn into a malignancy. So in the case of a heart attack, or an MI, the medical term, I just wanted to show you a picture of the coronary arteries of the heart. So these are the main arteries. So when someone's having an MI, there's a lot of different chemical processes going on in the heart. I'm just going to highlight a few. So the main part of the heart right here is more of the left side. So let's get here. So you have the left coronary artery. This right here, the left anterior descending artery, is one of the main arteries. So if this is completely blocked off, then you're going to have death to this entire side of the heart. The right coronary artery is more of the back of the heart. There's obviously also other arteries that stem off from that, but just to give you an example. So there's a couple things that are going on here. You're having some cell damage. So when cell damage happens, remember, we all have cells, and especially the lining of the artery. So what we have commonly used to think is that you would have an artery here, and over time you'd have cholesterol build up, then it'd build up more, and then build up, and then you'd have a heart attack. But that's not necessarily the case. It's actually rare that happens. You usually have some unstable plaque in the lining of the artery that is unstable due to maybe smoking or genetics, lifestyle, alcohol, and that causes a plaque to build up here. Well, what can happen is that plaque can become unstable and a little piece can break off. Well, if a little piece breaks off, it's your body's mechanism, defense mechanism to rush platelets to the area and try to heal this area, kind of like getting a cut on the outside of your body, and then it ends up blocking it off and you end up having an MI. But let's go deeper than that. So what happens is lactic acid starts producing, and that produces from pyruvate. We have pyruvate in our cells that helps with our metabolism. But then if we have lactic acid being produced, remember our pH is supposed to be between 7.35 and 7.45 in order to maintain, oops, it's not drawing here. It's in order to maintain homeostasis. So if you're not getting oxygen, that pH is going to change. And the pyruvate's going to excrete lactic acid from in the cell, and the pH is going to go down. And that's a more acidic environment. And what happens if it's an acidic environment? You have inflammation going, as well as what happens in the cell. Sodium and water start moving into the cell. Let me erase this. Oops. Okay, sodium and water, you have the in the cell and out of the cell. And if too much are moving into the cell, what's going to happen? The cell is going to swell up. And this causes a lack of oxygen, meaning hypoxic cell injury. So the movement of sodium and water going into the cell, it's not balancing out. That's a hypoxic cell injury. Another term that you can call it is the sodium-potassium pump, because if sodium and potassium are moving in, the potassium is unable to move out, and the organelles swell, the cells swell, and the sodium-potassium pump basically is not working. So this is what's happening when there's destruction or not blood going to one of these arteries. So let's say this patient goes to the cath lab and they go ahead and open it up with a stent and they restore blood flow. It does not mean the damage is finished and everything is back to normal, because then at that point you can still get damage to the cells, because now your body starts producing free radicals, which is that can be toxic to your system, and it's not normal. So you're not supposed to be excreting a lot of free radicals, because what it does is it can cause cell damage. It binds to all the cells and ends up causing damage. It's not meant to be there. Okay, to talk about intracellular and extracellular space. So we have sodium and we have potassium. We have chloride. Remember, sodium chloride go together. There's also other ones which we'll learn more about, but let's just talk about the intracellular and extracellular space. So when we think of intracellular, that's inside the cell. There's supposed to be a certain amount of concentrate versus outside the cell. So where is the largest percent of water in the body located? They're inside the cell. So let's think of someone with kidney disease. They're not able to filter out all the solutes and the sodium. That what happens is the intracellular fluid starts going to the extracellular fluid, and as well as what we call third spacing. And we get a lot of swelling in the lower extremities, especially. So a normal homeostasis is the intracellular fluid. But what happens is if you have a normal flow of solutes going in and out of the interstitial space, that's called hydrostatic pressure. And that is normal, but when you have an abnormality such as a failure of kidneys to filter, that hydrostatic pressure does not work as properly to keep the fluid in and out of the cell, so that fluid ends up going out of the cell, or it can't get, the other way will happen where you can't get fluids, the sodium and water, back into the cell, so it sits out of the cell. That's why we have to give diuretics, such as Lasix, to patients. This is the same thing with congestive heart failure. And when we go back to osmolality, remember that's moving across the membrane with passive transport. Molality. The most important solute is sodium. So we have a lot of sodium. Remember, sodium and water, they attract each other, basically. Where there's sodium, there's water. So that is the most important substance that determines the osmolality, because there is so much of it and it attracts water. So, for example, if somebody has a lot of salt and eats a lot of Italian sausage or pizza, and you're not drinking enough water, you're gonna retain all the water that you have in your body, and sometimes you wake up with puffy hands because all that sodium is attracting as much water as you have. That's why you have to drink more water. It seems like it'd be counterproductive, but it's really not. You drink more water, you're gonna flush a lot of that sodium out. So when you think of somebody then on the opposite side that has a low sodium, what do you think is going to happen in these cells? So if you have a low sodium, the cells inside are going to have more fluid. So you're going to have an increase of sodium and water. If you have high sodium, then you're going to have, you're going to lose all that intracellular fluid and it's going to go out, if that makes sense. So these are the main things I want you to focus on. If you can go over your PowerPoints again and just review them. But this video here is very important for studying purposes, as well as take all of this, and if you're not sure, dig deeper into your textbook. And I will also be going online at one point to answer your questions if needed.